So I could modify my answer to you, Sarah, a little bit. Where the A is in terms of the middle sequence does matter. So again, this is a conservative. It's always usually like 20 to 40 base pairs upstream from the 3' AG. We find that A. Things like that. But I don't expect you guys to, that would definitely be advanced genetics topics to think about how the genome is organized. Yeah. All right, so last kind of, so we're just kind of drawing out the basics, what the features are of a transcript that are going to be involved in splicing. And then I'll walk through some, again, this is a kind of basic steps. Your book goes into a lot more detail and talks about nucleophilic attack and what atoms are actually undergoing this chemical reaction. But for a couple reasons, one, that's sort of a cut point for in terms of basic genetics, Bio 311, and more advanced genetics. And then another reason that we're not going into quite that depth of molecular detail in the mechanism is this is definitely an area where there's a lot of debate and not necessarily agreement about how this exactly happens. So that's cool and exciting, but I don't want to teach you guys something that in a year from now you'll be told differently if you go on with your genetics courses or you go to graduate school or anything like that. So it's definitely an area that's still being actively researched. But the other player that I wanted you guys to know about, so here's our transcript, and it continues like this on either side, right? Where most transcripts aren't 2 exons, okay? This would be many exons, many introns. Each of our introns have these GU, AG sequence. And then the other, I'll draw it in a different color, the other component that's going to be involved are called, well, geneticists think they're cute and they call them SNRPs. Okay, so it's SNRNP. What this stands for is small nuclear, let's see, ribo. So they're essentially, it's a RNA and a protein, kind of like a ribosome. It has RNA components and protein components. So it's a ribonucleoprotein. So SNRNP. So these are small, made up of RNA and proteins together, and they're catalytic, they're enzymatic. And so these SNRPs, SNRNPs, are what's going to make this splicing happen. Okay, so let's attempt to draw this. So what we have here is our transcript with 2 exons and 1 intron. Our intron has the GU, the A in the middle, and then the AG at the 3' side. And the first SNRNP, so it's the first SNRPs that recognize these splice sites and the branch point, they're going to sit, the first one will sit down at the branch point, and the other one sits down at the 5' in that GU. Okay, so you'll have a and I'm kind of trying to draw them like behind it, but you basically have your SNRNPs. They're all named like this is U1 and U3. I don't expect you guys to memorize the names. Okay, just know that they're these SNRPs that are RNA protein components that bind to that branch point and they bind to the 5' end of the splice site. Don't worry about memorizing the U1, U2 part. Okay. What happens is other SNRPs actually then recognize and bind, this looks like a weird, but the AG up here, and they're going to bring the ends together, if that makes sense. They're going to fold that intron, they're going to make a loop structure out of it by their affinity to each other. Okay, so these RNA protein components will actually come together so that what you end up seeing is more like this. So that intron gets looped up because those 2 RNA protein components pull it together.
So is there one for both the 5' and 2'?
Yeah. Yeah. Yeah, and I'll draw that here in a second. I wanted you guys to know the first two is at the branch point and the 5' end. They sit down and bind and recognize each other, and they recruit the other ones over. Yeah, but I'm not too concerned about you guys memorizing the names and that kind of detail. Okay. So now, and there's multiple of these. Okay, so they're all bound. The first two recognize that branch point, 5' GU, and then the others will all cluster kind of around here. I know this is like probably terrible looking from far away. But essentially what they're doing is kind of grouping this whole intron together so it makes this nice loop structure. So still our 5' 3' end. And what they're going to do specifically is the first cut is going to be here. Okay? So the first cut happens at that GU splice site, the 5' splice site. And again, this you can read in your text if you really want to see what they are hypothesizing is the actual biochemical reaction behind this. But again, this is sort of still somewhat in the theory stages.
Okay, so do they make a loop between the first splice site and the...
Not yet. They're not. So they're not. So the intron is not interacting with itself yet. It's just a loop. So it's just instead of having a flat, you went like this.
So the SNRPs actually...
The SNRPs.
And nothing to do with them?
Yes. What was your original question? I'm confused too.
So if there's not one at the beginning and the end, like how do they make a loop from the beginning to the end?
I think there's not one. You mean the SNRPs. They are. There is one at the beginning. There is one at the end. Yeah. They start by first recognizing the branch point and that 5' end, and then another one will come and sit at this 3' end.
Well, it's the one.
Sure. Yeah. So yeah, it'll come and then they pull it together. But what I didn't want you to be confused with, because what happens now if you make this cut here is this will interact with itself. It makes it's called a lariat structure. This whole mechanism, often you might even see it, instead of being called a pre-mRNA splicing mechanism, sometimes it's called the lariat mechanism or lariat. Most people nowadays don't know what a lariat is. I'm guessing not many people. Lasso, right? Yeah, lasso, yeah. We're talking about furrows, because we're talking about sand furrowing when we were doing rescues and stuff, and I was like, if you told that to my undergraduates, I bet you half of them wouldn't know what a furrow is. I went to school at UC Davis, I know what a furrow is. You know, this is farmland. Yeah, lariat, you know, cowboys. They have the lasso lariat structure that they swing over their heads and hog tie cows and cattle and whatnot. So lariat is this like rope structure, right, like that. So it's named because the intron makes a little lariat. And so when Brandy asked about interactions, I wanted to make sure she wasn't asking about that. And let me draw this bigger so that. So if we think about this happening, and what interacts is that middle A site that was up there. It gets looped around and interacts with the U from the GU splice site, right? Because A and U can hydrogen bond together. So this is now, let me give you, this is your 3' end, what was the 3' end of the intron, and the U's up here at your 5' end. And after you make this cut, it actually loops back on itself and is interacting with itself. Sorry, I didn't draw this like squiggly still. This is still squiggly. I don't know if that helps you or makes it worse. But that's all intron. And the exons now, So both ends get cut. Your first splice site is at the 5' end. It will loop back, and that U will interact with the A. And then the 3' end of the intron gets cut as well. Now you have two fragments. You have two exon chunks. You have exon 1 and exon 2 that need to be ligated together. And then you have this lariat lasso thing that also has
And that's the A.
all those snurfs still attached. Yeah?
That's the A of the branch cord, right?
Yes. Yeah. This A, that's our branch point A. The one we started with back here. So this is why it's a lariat too, because you still have, and maybe my scale isn't super accurate here, you still have quite a bit left over of the rest of the 3' end of your intron, because you're just taking this DU and connecting it up with the A. You're making a little loop for the tail. Yeah.
There was a homework question. It asked which of those proteins was responsible for holding the complex together in that configuration, and it said the answer was the fifth one, which is attached to the 3'. Is it saying that just because that one is?
Yeah, there's one that's down here that kind of not black. It attaches this like whole thing.
Yeah.
Yeah.
I was confused why they weren't saying the whole complex was together. They just said it was that one.
I think there's, yeah, so I think they're trying to get you, so since the textbook goes into all that detail on the mechanism and is actually giving you the names of each one of the snurps, they're trying to get you to think about a function for each one of the snurps. Like, so in this case, you know, if you guys were responsible for knowing that this is U2 is the one that recognizes the branch point, U1 recognizes the 5' end, U5 is the one that's holding it together. Yeah, yeah, which is, it's accurate, it's correct. I was just trying to make your life easier, but maybe that doesn't make your life. I don't know. Sometimes I think people are more confused when I just say it's just the black box and it happens. Just be okay with it. Just accept. But I don't know. You guys can let me know if you want more detail than I do.
No, it's fine. It's perfect.
Yeah. No, I know. Yeah. Yeah. And so I think the book is trying to get you to think about what's the function of each individual snurf. But they are working together as a complex. Yeah. And they're all still, what I'm trying to just show here is, yeah, they're all still kind of stuck on this lariat when it comes up off the transcript, and it goes away. Okay, so the intron will be degraded, and the snurps disassociate and fall away. So that's kind of your final, what ends up happening is we have this nice, just exons together, and then the intron and the snurps will move away from that area. Yeah.
I guess, so where is the second cut that's being made? The first one here by the 5, is it gonna be closer to the 3' end?
Yeah,
It's not repeated about where the AU is connected.
It says that 8. Yeah, okay, add your AG in there. Yeah. Yeah. So those splice sites are part of the intron, so they go away with the intron. They're not stuck on the exon. So it'll always cut to the 3' side of it, or to the 5' side of it, whether it's on 5 or the 3.
And it's G7, so the G.
Yes. Accurate? Yeah. This is good. It keeps me like accurate in my drawing. 242 students just accept it when I draw giant cats that couldn't ever exist in real life.
It's in the corner of my house. También me acordaba de esta. Oh, sorry. I didn't hit you on purpose. It happened.
Okay. Any other questions?
No, sí.
And the SNR and Gs, those are degraded as well.
No, they can be recycled. They can be, but they disassociate. So intron is degraded, and then your small nuclear, ribonuclear proteins. It's more that they disassociate. they disassociate and they can be used at a different splice site. Because if we have a gene that has multiple exons and introns, right, we need to keep doing this process over and over and over again, making these little loops and cutting them out along the whole length of the transcript. So these will be, yeah, they're not degraded, they disassociate from that lariat intron loop so that they can be reused. So that you could just reuse here. And this whole kind of process here, this is occurring inside of what's called a spliceosome. So in terms of where this actually takes place, and it's considered an organelle, which is a little weird to think about. But these, I'll say within organelles, that are called spliceosomes. And what a spliceosome is it's just those SNRs plus some other proteins, right? Because it kind of should be automatically clear that we need to do something about these two exons floating next to each other. You need some kind of enzyme that acts like ligase. You need a way to bring together those two ends. So you need additional proteins besides just the SNRs. And so this is all happening inside of a special area. And the spliceosomes are actually, this is kind of odd, one of the most abundant organelles in the cell. So this is obviously a really important conserved process that needs to be happening fast so that we can get moving on to translation and make a protein from your transcript process. So it's not the most, but one of the most abundant organelles in the cell. Everyone comfortable so far with splicing? Okay, we brought up splice variants. And splice variants come from, I think Melanie said the word there, but alternative splicing. This is kind of an extension to our discussion about splicing. So this is a variation in the patterns of the exon, or the order really. And we see alternative splicing occur in different cell types. So you can take the same gene, I can't think of a specific example of a gene, but you could take a gene that's in your epithelial cells and the same gene, say, in a muscle cell, and you might see splice variants between the cell types. So in terms of like where you would see this variation, you would see it potentially between different cell types, because they might need a slightly different protein depending on how they use that protein for their cell function, right? Because all the cells have different jobs in our bodies. And so they might require a splice variant for their protein. And then another point is you might see this occur at different stages in development. And so again, thinking about what's the actual function of the protein, how is it used or needed at that point in development or in that cell type. And this is some really cool stuff. A lot of focus has been done on this recently, especially because, as you can imagine, if folks were only characterizing proteins from a certain cell type at a certain stage of development, and they thought, this is it. This is I've characterized my GFP protein. I don't know, whatever your protein of interest is, this is my protein. I know exactly what the sequence is. And then all of a sudden, someone decided to purify that protein from a different cell type in the same organism, but different cell type. were at a different stage earlier in development or later in development. And all of a sudden they're seeing a totally different amino acid sequence, but it's still functional protein, or it's still present in high levels in the cell. And I think that initially when people first discovered alternative splicing, it really threw people off because they thought, well, we were wrong here. This is the wrong amino acid sequence, when in reality it's still the right sequence. It's still the same protein, same name protein, but has, now we're seeing these transcripts get spliced differently. Okay, so it really opened up people's thinking about where and when to sample to do gene expression studies. Okay, because depending on the stage of development of the cell type, you could the tissue type, you could totally change what kind of variant you're actually sequencing. I know a lot of you guys know I worked in a chicken lab, and one of the other, our lab focused a lot on telomere biology, and so I worked on a virus, but other graduate students and postdocs that were in the lab did a lot of work on TR and TERT, so the telomerase RNA. And one of the gals, she found, I think something like, this is totally a side note, so she found something like 19 splice variants for TERT just in chickens. So looking at different stages of development, different cell types. So 19 different forms of this protein, telomerase RNA. So it's really, to me, interesting to think about. There's your favorite protein that you thought you were so comfortable and knew so well, and all of a sudden you introduce this idea of alternative splicing, and you have a whole bunch of different possibilities out there for variation. So that's as far as she got, was just characterizing, like, holy cow, there's like 19 of them, and we still don't know really what they're doing or why they have these variations, and that's kind of further work. But you can imagine if you're trying to target using a certain sequence for only one of these 19, you could be missing the other 18 that are still in the cell with just a different sequence. Also, under your alternative splicing, there's something called trans-splicing. And this just means that instead of variations in the pattern for sequence of exons, this trans-splicing is when you're actually you're fusing exons from totally different transcripts. Okay? So exons on different pre-messenger RNAs essentially are ligated together. So that gets even weirder, but it exists. It's rare, but it's been detected in cells. So now we're talking about totally different transcripts, totally different messenger RNAs that the splicing machinery is grabbing an exon from one and an exon from another and ramming them together. So you're really switching up what will ultimately be a different amino acid sequence. Yeah?
So at this point, is our definition of a gene something that leads to one pre-mRNA, or?
Define one pre-mRNA.
Like one molecule?
But you need because right now we're saying that variations of the pre-mRNA, right, we can slice it differently and that's still a gene.
So a gene is a protein product, or?
Not necessarily, because we call tRNA and ribosomal RNA genes. Yeah.
So that's my question. What are we calling genes?
That's a good question. And you'll see a lot of different definitions. Yeah. There will never be ambiguity on an example. If I know that that's not where your question's stemming from, but for the rest of you, if that's causing alarm bells, I will tell you when it's supposed to be a gene or what a gene is. But that's actually a great kind of philosophical argument type question in genetics. What defines a gene?
So what
And people have different definitions.
Yeah, what do you, give me one.
Well, okay, so a real basic old school one was the central dogma. That's a gene. If it makes a messenger RNA, it makes a protein. DNA to RNA, it's a protein. That's a gene.
It seems like we're
But
moving
now
past
we're
the central
way
dogma.
past that. Yeah.
Because even in micro, we were talking about self-propagating proteins, just very briefly, you know.
Mm-hmm. But you're talking about prokaryotes too.
Yeah.
So yeah, so in terms of prokaryotes, we just talked about lactoferron, which totally deviates from you
Is that the
saying
breaking apart?
one messenger RNA. Yeah, this is polycistronic, so you have multiple genes coming off, I guess, off of one messenger RNA. I mean, how do you define? Yeah, so then the definition of gene becomes.
So then, would it be easier, what's the definition of a cistron? That's the smallest unit of,
A cistron is kind of synonymous with gene, so
Yeah,
it's
that's
sort
what
of circular. Let's get anywhere without arguing.
Okay.
Yeah, no, it's a great question, a great thing to think about. But people are really, and that's a big changing part. And you guys are kind of hitting genetics in a really cool time where there's been such a big change in technology and what we know, things like this alternative splicing. We're going to talk about RNA interference here in a little bit. There's tiny RNAs running around doing all kinds of crazy things in your nucleus that researchers had no idea about, 30 years, 40 years ago. This has exploded, in the last 50 years, I'd say genetics has come really far. So a lot of our hard-line definitions are no longer pertinent. You can take my genetics textbook that I used when I was taking a similar class to the one you guys are in right now, and some of the definitions are totally different now, and I'm not that old. It wasn't that long ago. They totally, redefine how we think about it. So yeah, I don't have like a good, Wikipedia probably has like a great just one-liner, this is a gene, someone who's a computer scientist probably wrote it, it's like nothing to do with genetics. Yeah, but it's a good question. Okay. We can stay on topic, I promise, somehow. Let's see, I have one more small piece to go, and actually, my small piece to add on to our RNA editing, RNA splicing, capping, tailing, all that gets us out of chapter 12, and then we'll move into chapter 15. So the last kind of thing for your notes after alternative splicing is a real short concept called RNA editing. And all this is, so speaking of all this new cool stuff that they're finding, which makes life kind of weird, is that the actual sequence of the transcript gets changed, like specifically, not changed in terms of exons, introns being moved around, but actual nucleotides can be changed after transcription has occurred. So often, the most common one is seems to be a C to a U for whatever reason. So a lot of cytosines get changed to uracil. This is very rare. RNA editing is rare and definitely still under investigation. But just so that you know that this is a possibility that can exist, yet another way for us to change the amino acid sequence, potentially, right, depending on whether this is a missense, a nonsense, or silent change, we could end up with a different protein slightly. That was all I wanted. I just want you guys to know it exists. It's called RNA editing, and it means that you're actually changing nucleotides, usually point mutation like that. Yeah.
and so they definitely know that it's being regulated somehow, it's not just like
Yeah.
the base is getting
Right.
changed randomly.
Right. So there's enough research that's been done that they know it's not an anomaly or a mutation, a one-time thing. They see it over and over again. And so far the data is supporting that it's mostly C's changed to U's. So there must be some beneficial, there's a lot of research that's in that area. Okay, so we're at chapter 15 now. So this is our gene expression control. This is where we're at in our scheme of things. So still same kind of concept, thinking about how to regulate genes, whatever we're calling a gene at this moment in time. But ways to change expression level of genes within eukaryotes. And the first kind of subject area or topic that we're going to go through in chapter 15 is about transcription factors. So we've talked a little bit about them in terms of initiating transcription, right? We have the TF2D and H and G. You guys need to know the functions of those. But we didn't really, it's kind of an odd, I think you hear a lot about transcription factors in other classes and in this class, but we haven't actually stopped, which is sort of out of sequence in terms of your pedagogy and learning this, but we haven't really stopped and given a general definition for them. So what is a transcription factor? What are they doing? You'll often see them called TF, right, something. So we already talked about the TF2 blank that are involved with starting transcription. So these are all regulatory proteins. So most of the time we associate transcription factors, these types of proteins, we associate them with activation, so turning on gene expression. So most of the time they activate and rarely inhibit gene expression. But there are a few examples of where they inhibit. So loosely speaking, they mostly activate transcription of DNA. How do they do this? How do they interact with DNA? Generally,
By binding and conformational change.
Yeah, so that's correct. They just, physically, these transcription factors, whenever you hear a name transcription factor applied, think about it being a protein that's going to physically touch and bind to DNA. So these guys bind directly onto DNA at specific sites. Okay, so they recognize certain binding domains. And the cool thing is these domains, so these sequences on the DNA, the actual transcription factor it's something like it's 10 to the 6th fold higher affinity. So the protein, the transcription factor, the TF, its DNA binding domain to that specific sequence has a 10 to the 6th fold higher affinity than it does to any of the other sequences on the DNA. So it's not, I know that was a concept for me that was a little weird to think about. Why doesn't a transcription factor bind over here or over here or over here? I mean, obviously it's recognizing a certain sequence, but because of the chemical interactions, it has a way higher affinity to bind in its specific spot that it recognizes. Okay, so these proteins are very good at binding the DNA. And then along those lines, in terms of how they're classified, so it has to do with either the sequences that they're recognizing. So this is, you're kind of making you back up here a minute so that it's not confusing. I'll put numbers because that seems to like keep me kind of on a track here. So what we're doing is we're kind of making a list of the characteristics of a transcription factor. So what is a transcription factor? We know it's a regulatory protein that binds to DNA, and we know that they're classified. And so they're put into categories, because there's lots of them out there. They're grouped, usually into families. And it depends on either their conserved sequence, or based on their three-dimensional structure. you'll see, what do we mean when we mean protein three-dimensional structure? This is old stuff from your basic molecular class. What kinds of 3D structures do we see in protein?
Structures.
What's the actual quaternary structure? So people are saying tertiary, quaternary, and then what did we call those quaternary structures in Emily said one of them? Yeah, so beta pleated sheet and then the alpha helix. So you'll see them called like a helix, turn, helix. This is just an example one, but they're being named based on their three-dimensional structure. There's things called zinc fingers. So these are 3D protein structures that you guys maybe kind of remember-ish. So they name these transcription factors based on what they look like. And the reason I'm also bringing this up, not just on the classification point, but their actual three-dimensional structure dictates what part of the DNA that they bind to, right? So this structure, this 3D structure, is going to dictate where on the DNA that they bind. So DNA has two different groove sizes, right? We haven't gone into like tons of detail on the structure of DNA, but anybody remember hearing it from other? Yeah, you have a major groove and a minor groove on the DNA as it makes that helix turn. Okay, there's all kinds of physics and chemistry behind the actual structure, the double helix of DNA, and there's a major groove and a minor groove. And these proteins, they bind in depending on their actual three-dimensional structure. They can either fit in the minor groove or in the major groove. So this classification is kind of important in terms of how these transcription factors function, where they bind in on the DNA.